Hi, I'm Mike Oliver. I'm the Sample Preparation Product Manager at Thermo Scientific, and I'm going to be talking today about the benefits of the next generation Thermo Scientific solar microelution product and how it can bring benefit to the bioanalytical fields within the biopharma and the pharmaceutical markets. Sample preparation is obviously a fundamental part of the workflow within bioanalysis. We're dealing with very, very complex samples. There's a lot of components in there that cause issues for us in terms of our analytical procedures, such as phospholipids. So these are major considerations. They're something that we really need to remove from the process because they cause considerable issues in terms of ion suppression, contamination of our column, so the longevity of the columns, the injector cleanliness, and obviously endpoint mass point detection. Maybe getting to the source, we need to clean our source more often, etc. So there's those factors, but obviously the quality of the data is of fundamental importance. And of course, there's obviously the issues of higher potency drugs. So we're looking at more and more drugs of this nature coming into the marketplace. So we have drugs at a lower concentration, eliciting a desired response. And all that is doing really is pushing down our sensitivity requirements, our quantitation limits further and further. We've really been working to address those needs from a sample preparation side as well as the other side. So if we look at the sample preparation side, one of the main products that we brought to the market to address those needs is the solar microelution product. So this is based on next generation SP design. It encapsulates embedded particle technology where essentially we remove the need for frits, which are present within conventional SP devices, and also develops a solid porous unit. So a lot of that design mitigates against the problems that we've seen with conventional SP, such as voiding, channeling, inconsistencies in packing. And we understand from our customers that reproducibility in sample preparation is a major issue for all of them. And if they can improve the reproducibility in that area, then it's a major benefit to them in improving their throughput and return on investment. Solar, because of that new robust design, it has less failure rates. We're able to process samples far quicker, but it also has some additional benefits. The microelution format allows us to extract samples in around 25 microliters, so that means that we can remove the blowdown steps. The blowdown step itself has a number of issues. One, it adds a considerable amount of time and cost to the overall process. And in addition, if we're looking at, for example, peptides and proteins, they're susceptible to loss during that phase. So if we can remove that process, then it's a considerable benefit to our customers. So that's one thing. The other thing is that we're able to load a large amount of volume and pre-concentrate it. We can do a 20-fold pre-concentration step, so load a mil of sample, elute in 25 microliters, and we get a big increase in sensitivity without having to adjust MS sensitivity as well. So that has a considerable benefit when we're looking at some of these higher potency drugs. So there's some major benefits that the technology brings to the fore there, and we're finding that our customers are bringing this technology into their standard workflows. It's benefiting their workflows. They're seeing increased sensitivity, increased reproducibility, but it's benefiting the entire workflow.